So today the title of my talk is basically metabolic adaptation in muscle during cancer. And I choose this title because I'm actually going to talk not only about the heart, but also the skeletal muscle. And as Ibro mentioned, I'm a new recruit at the Smith Heart Institute, and I'm going to be joining Cedars-Sinai at the end of this month. So if there are any trainees out there who would like to join a new lab, please, you can send me some notes afterwards. And so let's see if I can advance. No, doesn't want to advance. Now it wants to advance. Okay, great. And so my work, I'm really focused and interested in how does the heart basically as a complex biological system is able to adapt to various different physiological and pathological conditions. And whenever I give a talk about my work, I like to show this picture, which is basically on the cover of a book from Hachachka and Sumero called Biochemical Adaptation, and broadly covers all forms of biochemical adaptation in biological systems, not only mammalian cells, but also other cells or animals. And the heart is very much like what is shown here on the book cover, Chimera, which is able really to integrate all forms of stress and information and then adapt to it in a new environment. And this adaptation requires really an orchestrated response at all levels of the system, coming from the epigenome, the transcriptome, down to the proteome, into the metabolome. And me, I'm particularly interested in how all those stress factors or alteration can then lead to changes in metabolic flux and basically lead to a crosstalk between those different levels of the system, because metabolic changes are dynamic, they're not static. And so we need all those information to fully understand how large complex biological systems are adapting and responding in the context of a disease. And in order to study this experimentally, what I'm using in my work is really a systems biology approach. And there are a lot of people have different definitions for systems biology, but basically we are looking at the entire system or the entirety of a cell with all different all those different levels that I showed in my previous slide, by integrating various forms of data coming from all sorts of technologies. And you can use mass spectrometry, sequencing, but basically we're trying to interrogate all those different levels. And in my work, I predominantly use stable isotope tracing in vivo mouse models, but also down the line, we are aim for translational studies in human, where we are then able to trace specific metabolites and understand using mass spectrometry, either gas chromatography or liquid chromatography, the carbon distribution at a given time point. And taking this data, you can then integrate that into large-scale networks to understand really the flux distribution just coming from very few parameters. You will then be able, with computation analysis, to predict the fluxes in a large-scale network like a mammalian cell, and through that information identify specific pathways that are altered in your system. And so I apply those approaches to one big topic, which is basically the crosstalk between cancer cells and the heart. And so in basically recent cardio-oncology has emerged in recent years as a new clinical and research subspecialty, which is due to the fact that new therapeutic approaches have basically increased the lifetime for a cancer patient, but also showed us that there are basically increasing evidence for associations between cardiovascular diseases and also tumor diseases. And this intersection is increasingly recognized to extend beyond, of course, the cancer treatment that is known to perturb cardiac function, but there are also shared risk factors. Obesity is one of them, diabetes is another, but then also in a corresponding immune response and potentially factors that may be secreted by the heart that can make patients more or less likely to develop cancer. And there have been some very recent publications in the past couple of years by Catherine Moore's group and Afram last year in particular that, for example, show that in myocardial infarction and early hypertrophic cardiac remodeling can enhance the tumor growth. And so when we look at a subgroup of cancer patients, those so-called childhood cancer survivors, what recent data has shown is that due to the new development of new cancer therapies, overall the survival has increased. However, all those patients tend to develop at the later stage in their life during adulthood, when they're in their 30s and 40s, all of them have an increased risk for developing cardiovascular diseases. And this is really independent of the treatment. It doesn't matter if the patient received radiation, immune therapy, or classic chemotherapy. There is, depending on the tumor type, there is a higher risk for those childhood survivors to develop cardiovascular diseases. And this can range from 8 to 15 times. And based on this data, we are now looking more and more into the possibility that beyond the cardiotoxic therapies, other factors, basically the biology of the tumor, may induce remodeling in the heart, and understanding how those pathways are activated, how the heart is remodeling in this patient population may also help us identifying not only targets for treating these patients in a better way, protecting the heart during their treatment, but also provide us potentially mechanisms for other forms of heart failure that are independent of cancer, and allow us to really use these as common concepts in other pathologies. And so what I'm really interested in is how the metabolic phenotype of cancer cells can crosstalk with cardiomyocyte and lead to remodeling. And one specific cancer that I'm focusing on is the acute myelotic leukemias. And acute myelotic leukemias are very fascinating in the way that there are basically two different patient populations that we need to look at. It's both patients that are very, very young, from 0 to 5 years, so infants, and then adult patients where the median age range is around 65 years. And those two patient populations are very, very different, both in terms of their survival rate, but also their genetic profile. And what is interesting in this is that in cytogenetically normal AML, about 10% of those pediatric AML patients show mutations and genes that are known to modulate the epigenome, which are highlighted here. One of them is the NDNMT3A, TET2, and IDH1 and 2. And IDH1, for all the aficionados here in metabolism, is of course a Krebs cycle enzyme, which is predominantly mutated in IDH1 and 2 cancers patients, and leads to a very interesting metabolic phenotype that I'm going to talk to you now a little bit more about. And so the metabolic reprogramming in cancer cells is really a hallmark that has been recognized now for many, many years. And the mutations that often occur in cancer cells, like gliomas in particular, but also the leukemias that I was mentioning, are the fumarate hydratase, succinate dehydrogenase, and the IDH or isocitrate dehydrogenase. And again, those are all Krebs cycle enzymes. And particularly when IDH2 is mutated, or IDH1, what that means is that the enzyme gains a neuromorphic function, and instead of decarboxylating isocitrate to alpha-ketoglutarate, starts producing a new metabolite, a so-called oncometabolite, dehydroxyglutarate. And dehydroxyglutarate, at least in gliomas and cancer cells, has been shown to lead to the inhibition of alpha-ketoglutarate dependent deoxygenases like TET2 or polyhydroxylases, and through that epigenetically modify the cancers and leads to a hypermethylation state inside those cancer cells, which provides them a benefit for proliferation and growth. And what happens in these patients is that we can observe a significant high level of the D-hydroxyglutarate in the millimolar range. And when D-hydroxyglutarate builds up, it quickly starts inhibiting those endogenous cellular systems through known biochemical or enzymatic inhibition. And what is now fascinating is, as an example, the overall goal, what I'm using in my work is really trying to understand how those tumor-specific properties are now leading to metabolic remodeling in the heart, initiate and maintain structural remodeling. And through that, we may be able to identify potential new liabilities and therapeutics using the knowledge of which pathways are leading to structural remodeling may provide us an information about common concepts in cardiac remodeling, but also might give us new metabolic targets beyond the ones that have already been identified. And this applies both to the heart, but also the skeletal muscle. And so the two projects that I'm going to talk to you today about are how metabolic adaptation occurs in the heart in response to D2-hydroxyglutarate. And in the second part of my talk, I want to show you what consequences this oncometabolic remodeling has on the skeletal muscle, and how essentially autophagy and the harnessing protein remodeling can orchestrate and drive remodeling in a muscle. And so what we did in collaboration with Peggy Goodell's group here at Baylor College of Medicine in Houston is we used an overexpression model in vivo where we overexpressed a common IDH2 mutation, the R140Q, which occurs in about 12 to 14% of the AML patients that I showed to you before. And we transplanted hematopoietic stem cells in bone marrow irradiated mice. And what we observed just after the induction is that the hearts of those animals were enlarged. And also at the molecular level, we found that the expression of myosin heavy chain alpha to beta was switched, which is very common whenever and the heart is remodeling and a heart failure is occurring. And so we had a reduction in the myosin heavy chain alpha to beta ratio. And these animals showed about 4 to 12-fold increase in the L and D2 hydroxyglutarate, suggesting that this may be driven through an oncometabolic remodeling because the hearts in these animals were still expressing a wild-type IDH2 form. And so we ask a very, very simple question: does D2 hydroxyglutarate as a metabolite impair the heart? And if so, how? And so, as I mentioned at the beginning of my talk, I try to look at the heart really in a comprehensive, holistic way. And one nice model to do so in a controlled environment is the working heart perfusion system, and which is shown here in panel A. And what you can do with this technique is you extract a heart from rodents or any other animal, and you can connect the left atrium and left ventricle with two cannulas, and through infusion of a defined buffer, you're then able to perfuse the heart in a normal circulatory mode. And in this apparatus, the heart is essentially beating, which is shown here in panel B. And as you can maybe appreciate, is that this is a really, the heart is beating at a normal heart rate as is found in the body. This is a rat heart. And we are able with this preparation to both perfuse the heart with a known defined composition of nutrients, but also tracers to understand what is the carbon distribution and which nutrients are being utilized and oxidized. And you can then also measure physiological parameters, cardiac output, coronary flow, oxygen consumption. And the drop that you see here at the bottom, that is really the coronary effluent, which will allow us to then understand which metabolites are released from the cardiomyocytes and from the tissue into the bloodstream. And with this assessment, we basically perfused wild-type red hearts with or without D2-hydroxyglutarate at a level that is found in AMR patients. And what we noticed is that even in just a very short-term perfusion at 10 minutes, the cardiac function declined rapidly. And then when we stimulated those hearts with epinephrine, after about 10 minutes, what we noticed is that the adrenergic response was almost absent, and the hearts were not able to maintain the increased work demand that we imposed on them, indicating that there is really a acute contractile dysfunction that was induced by the oncometabolite. And as I mentioned, D2-hydroxyglutarate is a structural homologue to alpha-ketoglutarate. And so the difference here is really just at C2, instead of having a keto group, there's a hydroxy group. And what we then tested is whether or not D2-hydroxyglutarate could potentially bind to alpha-ketoglutarate dehydrogenase and lead to an enzymatic inhibition. There are various ways how to test this hypothesis. The approach that we used is using a pronized digestion blot or a DARTS blot. And the idea behind it is if a small molecule can bind to a protein, it's basically able to stabilize such protein. And in presence of pronase, it is then less susceptible to degradation. And so with increasing concentration of that small molecule, we would see an increase in the bands on the Western blot in presence of pronase. And this is exactly what we observed. And so D2-hydroxyglutarate is a potential binding partner for alpha-ketoglutarate dehydrogenase and leads both to a reduction in alpha-ketoglutarate dehydrogenase activity, and that is acutely and concentration dependent. And so in the next step, I included all this data that we gathered through the experiments in an ex vivo setting into a large-scale network analysis. And so I developed during my PhD a mathematical genome scale network called Cardionet, which is depicted in this picture here. And Cardionet consists of almost 2,000 metabolic reactions and has assigned over 900 metabolites. And my goal is to make this network also more accessible to other researchers. So watch out for that. It's going to be part of my new appointment also at Cedars. But the nice thing about Cardionet is that you can use it to interrogate metabolic pathways that we are sometimes not able to measure experimentally, or that are a little bit too difficult to assess, and just with a few parameters. And one approach for that is when you look mathematically at computational modeling of biological systems, there are various different techniques. And one of them is, of course, kinetic modeling. So the idea here is that you use Michaelis-Menten kinetic to describe mathematically biochemical reaction. And so a simple reaction like, for example, the hexokinase, where glucose is phosphorylated to glucose 6-phosphate, can turn into a very complex mathematical equations where we often do not have all the parameters available for said enzyme. This technique is very can be applied to pathways like glycolysis or the Krebs cycle, but other more adjacent pathways are, and this is starting to be very, very difficult because we simply do not have all the information about the enzymes from the given biological system that we're looking at. And so one technique or another technique that you can use to circumvent this problem is called flux balance analysis or constraint-based modeling. And what we are doing in this analysis is we're simplifying our differential equation by assuming that any given biological system will reach eventually at a given time point a steady state. And so we can use this steady state assumption where the sum of all fluxes in a given system will reach zero to turn differential equations into linear equations that then can be solved numerically, even with using a large-scale network like CardioNet. And that's exactly what we did. And so we used the parameters coming from the working heart perfusions and integrated those into flux balance analysis simulations. And when you then visualize the fluxes, again, in a simplified way, what we observed is that in control simulations, when dehydrogenase is not present, we have glucose is taken up and decarboxylated in the glycolysis in the Krebs cycle, and ATP can be generated. However, in presence of dehydrogenase, as you may appreciate in this slide, the inhibition of alpha-ketoglutarate dehydrogenase not only leads to a reduction in the flux in the Krebs cycle, the flux is still present, but it's reduced, and an increased flux through the glycolysis. So what we observed in the working heart perfusions that the hearts were less able to beat were also corresponding with a reduced ATP provision through available metabolic pathways, indicating really, as we could show here through our computation analysis, that the predominant ATP provision was came from glycolysis instead of oxidative phosphorylation. So it was less efficient and required far more nutrient uptake and different types of nutrients than under normal circumstances. And one other way of interrogating this is using stable isotope tracing. Whenever dehydroxyglutarate is inhibited or there's a disruption of the Krebs cycle, there's a chance that those Krebs cycle intermediates can be redirected into other pathways. And one way to test this is with uniformly labeled glutamine and uniformly labeled glucose. If indeed there's a redirection of Krebs cycle intermediates, if you perfuse a heart with uniformly labeled glutamine, what happens is that in a reverse, and using the reverse function of the isocitrate dehydrogenase, isocitrate gains an unlabeled carbon, which then can be measured using mass spectrometry as a so-called M+5 fraction in citrate. And this is exactly what we found and observed in our perfused hearts, is that in presence of D2-hydroxyglutarate, we suddenly had an increase, and significant increase in actually an occurrence of a citrate that had one unlabeled carbon, indicating that the inhibition of alpha-ketoglutarate dehydrogenase through D2HD leads to redirection of Krebs cycle intermediates into citrate. And so when citrate is generated reductively, there's a chance that through transport activity, citrate can be exported from the mitochondria into the cytosol and then becomes available to other metabolic reactions like the ATP citrate lyase, and can be used to de novo synthesize acetyl-CoA or oxaloacetate. And correspondingly, what we have found in those perfused rat hearts is that really in presence of D2HD, there was an increased panacetylation of histone 3, indicating that D2HD is linking not only in cancer cells, but also in the heart, in cardiomyocytes, metabolic remodeling or stress with an epigenetic response. And this is something that I'm working on right now. We have another project where we're trying to understand what other mechanisms are involved in which specific gene programs are activated, so watch out for that. But I want to switch gears now and tell you a little bit about the other consequences of an increased availability of D2-hydroxyglutarate on a different muscle form, muscle type, and in this particular case, a skeletal muscle. And so another stress response pathway in cells is autophagy, and autophagy can be activated through various different signals, and nutrient stress or oxidative stress, but it's basically allowing a balance between protein synthesis and the breakdown in the skeletal muscle is through that regulated by various different factors. And this pathway can then be used as a protection against potential damage. And so during autophagy, cells from double membrane vesicles and autophagosomes that then sequester organelles, proteins, or other portions from the cytoplasm, and then are delivered to the lysosome and allow cells to essentially scavenge nutrients like amino acid, fatty acid, and glucose. And what we ask in our project is, if we have this response, metabolic response on the heart, is there potentially also a response in the skeletal muscle in animals, and what would be the mechanism for this response? And what we did is we incubated L6 myocytes, which is a red skeletal muscle cell line with our dehydrogenase for 24 hours, and then did a series of targeted bioassays, but also metabolomics analysis. And what you can see here in the PCA analysis of the our mass spectrometry assessment is that there's a real difference between the metabolome of untreated and treated cells. And what we identified is a series of metabolites that are specifically up or down regulated in vitro in those skeletal muscle cells. And in particular, these metabolites are part of amino acid metabolism, but also the NAD redox. And what we were then able is to include those into a network analysis, coming from the information from CardioNet, but also the mass spectrometry assessment that we did. And so the resulting network can be visualized with cytoscape, which is depicted here in this panel, and consists of over 100 metabolites with around 600 different metabolite interaction, which then allow us to identify in a more bird's eye view which pathways are really affected by dehydrogenase. And this analysis showed that the predominant pathways similar to the heart that are affected in skeletal muscle cells by DHD is the pentose phosphate pathway, the redox state. We really saw an increase in the NAD to NADH ratio, and also an increased accumulation of amino acids. And so we wanted to prove whether or not there's an increased activation of autophagy in presence of dehydrogenase. And there are no single autophagy marker that you can use. You need to use a various set of assays. And so we used both Western blotting, but also microscopy to assess whether or not DHD activates autophagy, in particular autophagic flags. And the first marker that we looked for was an LC3 expression. And what we found is that in presence of DHD, there's really an increased lipidation of L332, and that is with or without baflomycin. And when using GFP tagged L3 we observed that DHD increases the L3 punctate formation in redistribution from the nucleus that we usually see in untreated cells into the cytoplasm and into the cytosol. And those are co-localizing with the lysosome, really indicating that there's not only an increase in the LC3 into expression, but also flux. And recent publications have shown that SIRT1 as an NAD-dependent belonging to the NAD-dependent sirtuin is driving or can drive the deacetylation of LC3 inside the nucleus and redistribution of LC3 into the cytosol. There's a very nice paper from Huang et al., which was published almost 6 years ago, where they show that through nutrient deprivation, and there's an increase in co-localization between LC3, 13-1, and then a corresponding deacetylation of LC3. And because we observed this change in the redox state, which is induced by D2HG, we then tested whether or not there's also such co-localization occurring in our system. And this is exactly what we found. Using a chromium precipitation, we observed, and this is just a representative plot, we observed there's an increased co-localization between LC3 and 13-1. And correspondingly, when we assessed the acetylation of LC3, we found that at baseline, the acetylation of LC3 in untreated versus D2-hydroxyglutarate treated cells was reduced. And so we developed mutants of LC3, which basically are hypoacetylated, and where we replaced the lysine and arginine at position 49 and 51. And so those mutants are, as you can see in the control cells, are hypoacetylated. However, when we treat those cells again with D2-hydroxyglutarate, this acetylation pattern is even further reduced, indicating that yet again, there's an increase in flux and an increase in the activity of deacetylation occurring at LC3-2. And that is also correspondingly can be observed again in using microscopy and GFP tagged LC3, where we also found an increase in the punctive formation in presence of our oncometabolite, indicating really that D2-hydroxyglutarate is driving the deacetylation of LC3 and activation of autophagy. And so as a proof of concept, what we then did is we overexpressed a acetyltransferase P300 and knocked down 13-1. And what we expected was that there's a decrease in autophagy, and that is exactly what happened. With P300 overexpression or 13-1 silencing, we could attenuate the effect of D2-hydroxyglutarate. The cells were not necessarily healthier. So the livelihood or the longevity of the cells was actually quite bad, which points towards that essentially autophagy is needed in case of D2-hydroxyglutarate to harness all those amino acids and potentially fatty acids and be used to kind of counteract the energy depletion that D2-hydroxyglutarate is causing. But we think this is a potential mechanism that can explain how inhibition of a-ketoglutarate dehydrogenase can lead through to both epigenetic modification, but also then a proteome modification. And of course, this was all in vitro. So in the next step, we wanted to test whether or not our findings are also applicable to an in vivo setting. And so we treated mice for about a month, starting from the age 10 to 13 weeks with D2-hydroxyglutarate daily. And during those treatments, we also assessed their grip strength and weekly grip strength measurements. And we did this both on males and females. And what we found is that there was a significant reduction in the weight of the musculus gastrocnemius in male animals, but we couldn't really observe that in females. However, when we measured the grip strength in those animals, we found that both in males and females over the course of their growth, even when we normalized this to their body weight, we observed there was a significant reduction in the grip strength. And we measured this grip strength with a very simple experiment where basically the animals were voluntarily lifting teeny weights. defined weights coming from a balance. And whenever the mouse was able to lift a weight, it gathered a point. And so with increasing weights, we could then test whether or not the mice really had a growth-dependent increase in their muscle strength, or if this was affected by the oncometabolite. And so at the molecular level, at the next step, what we then did is really a multi-omics assessment of the molecular changes within in the skeletal muscle as a response to dehydrogenase treatment. And for that, we used RNA sequencing and mass spectrometry-based proteomics and metabolomics. And for that, we really needed to interrogate various different types of mass spec. It wasn't just one an LC system, but different types. And so anybody who would like to use this type of analysis, please I'm happy to answer any questions when it comes to that, and I highly recommend looking into different forms also of mass spectrometry to get a broad understanding of how the metabolome is changing. And what we observed in all three different types of data sets is that there was a separation not only between our treatment groups, but also between the sexes. Similar to our observation coming from the phenotype, the males and female animals that were treated with dehydrogenase showed differences in their gene expression pattern, in their proteome expression, and also in the metabolite expression. The females overall overlapped at those analysis more with the control groups than the males, which was very interesting. However, what we were then able to identify was essentially a subset of proteins by cross-correlating the transcriptome with the proteome information. And in analyzing those different data sets based on whether or not there was an overlap between gene expression and protein expression. And as you can see in this panel, there is a very poor correlation between those two parameters. And that is not unusual. This has been described in other studies under various other different stress conditions. However, in our particular case, it also indicates that there are potential post-translational regulation that may drive a different proteome pattern than we observe using gene expression analysis. And the other thing that we notice is that we specifically identified proteins that are part of again, amino acid metabolism. I listed here the proteins. Please feel free to look them up later, but Endufer is one example. BDH1 is a key regulatory enzyme in the amino acid branch in amino acid metabolism. And we further found also that basically forms of different forms of sirtuins were up or down regulated in presence of dehydrogenase. And when we then did another analysis where we essentially used cross-correlated the proteome transcriptome information with the pathway set information as a 2D enrichment analysis, we were then able to identify pathways that were differentially regulated in males and females. And this particular view was very important because it clearly showed to us that not only individual proteins, but also entire pathways, if you interrogate all the information coming from those large-scale data sets, were different between, again, not only the treated and untreated groups, but also between the sexes. And so the female animals upregulated proteins and genes that are part of the skeletal muscle contraction, and in particular, they upregulated the protein expression of proteins that are part of oxidative phosphorylation and the electron transport chain, indicating again that they're which links back to our initial observation that those animals were still able to lift their weights while the males were not, which is then also shown in the male group, which downregulated all those pathways, indicating that males and females had really a different strategy and response towards the adaptation to the oncometabolic stress. And when we specifically looked into which particular pathways are cross-correlating between the transcriptome, the proteome, and the metabolome in a multi-omics protein-metabolite interaction analysis in the male set, we identified basically 25 metabolites that are depicted in this panel that show ARBO downregulation at the metabolite level and a corresponding protein in the corresponding pathway. And with that, we were able to identify 25 metabolites and 49 proteins that are here depicted in a network and belong broadly to the pyrimidine cofactor metabolism, amino acid metabolism, Krebs cycle, and glycolysis, and explain why the male animals showed basically a different phenotypic characterization in response to dehydroxyglutarate. And so what we think is happening is that the inhibition of alpha-ketoglutarate dehydrogenase leads essentially to an increased demand in male animals in glycolytic flux, and increase in the NAD/NADH redox, and through that an activation of SIRT1 and activation of autophagy, and over time and remodeling at the proteome level, in particular in the branched chain amino acid metabolism, which leads then to a broad remodeling and loss of muscle mass. And in summary, what we think is really the redirection coming from dehydroxyglutarate and inhibition of dehydroxyglutarate of alpha-ketoglutarate dehydrogenase and the corresponding redirection in the Krebs cycle is orchestrating this metabolic remodeling and driving metabolic remodeling, and through that inducing a proteome remodeling. And what I want to focus on in my future work is really understanding which other potential signaling pathways are involved in driving this response and potentially can be used to prevent the phenotype that we have observed, and understanding and identifying potentially small targets that inhibitors that we could use in the clinic also to protect not only the heart but also the skeletal muscle in this patient subpopulation. And so with that, I would like to thank everybody who was involved in the study. Of course, my mentor, Dr. Techmeyer, and our very small lab with our two research assistants, Benjamin and Rebecca, and then everybody at UT who was instrumental in pulling off the mass spectrometry, and our collaborators at Baylor College of Medicine with Peggy Goodell, my new home at Cedars-Sinai, and of course UT Southwestern with Ralph Berardino's group, who is also a dear mentor and who helped me learning how to use GCMS and conducting stable isotope tracing. And with that, I like to thank for your attention, and I take your questions. Thank you very much.
Thank you so much, Anya, for this amazing talk. And we can now take questions. As you know, you just raise your hand. And yes, so I'll start with Mabruka. Yes, go ahead, Anya.
Really very great science and excellent way of getting everything by analysis. So I love that proteomic and metabolomic analysis. So I'm going to ask you very naive questions. Is about, seems like there is a balance between D2HG and alpha-ketoglutarate productions, the two arms. So what tell the IDH2 to go toward the directions of making more D2HJ in disease conditions? What's make the enzymes to produce more of that rather than going through the alpha-ketoglutarate, the productive pathway?
And so it's really a mutation. So the mutation that is, so the protein, the IDH1, is altered structurally in those tumors. And so so there are two forms of the 8-hydroxyglutarate. There's an L and a D form. And the what is now known is that both forms are usually produced under normal physiological condition, in particular the L form. And what happens is that the cells are usually able to degrade L2HD through another dehydrogenase, which is called L2-hydroxy dehydrogenase. It's a very long acronym, but every cell can convert D and L to hydroxyglutarate back to alpha-ketoglutarate. And what we think is happening is that through, again, enzyme kinetics, this reaction is also overwhelmed through the inhibition of IDH1. The 2-hydroxyglutarate dehydrogenase requires FDA, and so there's a limit enzymatically to how much the enzyme can, or the Vmax is overwhelmed, and the enzyme is saturated after a certain point. And in the tumor setting, this leads to the slow accumulation. And so in IDH1 and IDH2 mutant tumors, you see a variety of ranges of the D2-hydroxyglutarate, which is a little bit confusing, but I think the explanation is really how good or bad the system is balanced, and the corresponding dehydrogenase is actually able to convert the oncometabolite or the structural homologue back to alpha-ketoglutarate. And so in the preprint that you see that I cited there, in the supplement, what we actually also did in vitro is we tested various different concentration of the D2-hydroxyglutarate, and there is up to 0.1 millimolar of D2-hydroxyglutarate. The system in vitro, we do not observe any changes in mitochondrial function or cellular death or autophagy activation. But beyond that, there is a gradual and concentration-dependent increase in those in the phenotype. So there's a reduction in ATP provision, there's a reduced mitochondrial function, and that is really linked to the dehydrogenase activity. The Km value is, at least in the literature, reported at 0.13 millimolar, and so that all very nicely fits. And if once we overwhelm the system, there's really, it's beautifully biochemistry and enzyme regulation that is occurring. And so that's why we think also, or others have also already thought about this with those IDH1 and 2 inhibitors, that you could push the system back to a normal physiological level by disrupting this overproduction. And this can work, but there's still more data and studies that need to come out. And yeah, I hope this answers your question. There's a lot more to it, but yeah, this is my short answer.
Thank you.
Thank you, Mabruka. And Thomas Pulnico.
Oh, hi. Thanks, Ebru. A very nice talk. A quick question regarding the metabolites' effect on lysosomal function. Is there anything known, or have you probed whether these intermediary metabolites have any direct effects on the lysosome function, and also do they affect any of the transcriptional regulation of lysosomal functioning?
Yeah. So there. To the first question, beyond what I have shown you in terms of just measuring the LC3 lipidation and doing the microscopy analysis, we did not specifically look for other lysosomal markers. But in the gene expression analysis and the in vivo setting, we also did a proteome analysis on the in vitro data so basically at time point 24 hours and then after 30 days, there's no proteome change after 24 hours treatment with dehydrogenase. We have several lysosomal markers that are popping up. So we had RAB7 and FOXF3 that were altered in the 30-day treatment, and there are several others that I would have to look up in the data sheet, but out of the top of my head, those ones I can tell you, indicating really that there's the delayed autophagosomal lysosomal fusion is affected for this, and LAMB1 as a marker was also increasingly expressed at 30 days, and there's also a difference between males and females. And again, we have that data in the preprint. We highlighted those, but there are some others. And so the several RAB proteins are differentially expressed in those groups at 30 days, not so much after 24 hours. And that's why we also think that this is an acute metabolic, like the metabolism is driving the response in the acute phase, but then over time, gene expression programs and other signaling pathways are driving and defining the phenotype in a more chronic setting. But the initial stress regulation is done metabolically.
Thank you.
Yeah, you're welcome.
Jason.
Hi, Jason.
Hi, Anya. I really loved your talk. Thank you so much for sharing.
Yeah.
I have lots of questions, but maybe I'll just limit to one or two. So, you know,
email.
you know, I'm sure in your experience, you know, one things you've encountered is that oftentimes you know, when we do metabolomic profiling, we're quite limited by the number of standards that we have, right? And so sometimes what we get is we get spotlight effects where, you know, the platforms or the standards we have are overrepresented in central metabolism or amino acids, nucleotides. But we know from physiology that, you know, cholesterol, lipid metabolism, right? There's several other aspects which play important physiological roles. So my question is how you kind of deal with thinking about, you know, I know the models are useful for predicting things, but they're only limited by the constraints. So I'm wondering, you know, how you think about handling or dealing with kind of the things that you can't measure. And then second, you know, the comment that you made, I really love that clustergram you had of central metabolism, amino acid pathways, et cetera. You know, in thinking about, you know, the tension between how you think about the tension between some processes being pathway specific or, you know, common effects of insults to a shared pool, like as an example, energy currencies, right? So, yeah, so thank you.
Yeah, no, thank you. I think you're hinting on a very, very, very important topic, both specifically for science, but then also philosophically. Because you cannot know what you don't know. And it's one of those things about mass spec, when you're dealing with mass spec, in particular with proteomics, from a technical standpoint, anybody who has ever done or received proteomics data, the people will tell you there is missing data. We will have information that is lost because of the way we are measuring metabolites and proteins and sequencing. Technically not so much, but how do you deal with it? So my approach, that's why I'm so much into systems biology as a way of you use different types of techniques. And you may have noticed I'm going almost through the entire field of the molecular biology techniques. I do sequencing, and we have done everything short of SHIP-seq, but actually we did that too, but I have not the data presented. And my approach right now, and I'm not sure if it's the right one, is to use various different types of techniques, combining both large scale data set analysis with targeted analysis to just cover as much as possible and eventually have a Venn diagram that gives me a full picture. And you will never reach perfection, but we can thrive to it. And I think that's where the computational analysis, when used correctly, can be very, very helpful and powerful, because we can use, and that was kind of what I was trying to do with those network analysis, to use metabolites where you can have hundreds of data, with proteomes thousands, and with sequencing you have tens of thousands of data points, to combine this in a human digestible mode. Because in the end, we are scientists, we have to look at that Excel table or at that picture and then make a decision. Are we going to put ourselves or the postdoc or the grad student onto that next project and tell them run that Western blot? And so the pathways are a construct. And I think this is very, very important to understand, that this is a human construct that we came up with to make complex systems easier to understand. And as long as you know about that limitation, you're never going to, you're going to be aware that your data is limiting. So this is kind of where my philosophical approach comes in. But technically, what I would like to show with this study also is what you can do with sequencing data, proteome and mass spectrometry data, go beyond the top hitters, because just because a metabolite or a protein goes up or down does not necessarily tell you anything. A stable system that reaches a steady state may have not any changes in a certain given metabolite, but the surrounding periphery is altered. And by using this network approach and by looking at trying to have these integrative analysis, whenever you're getting sequencing or mass spectrometry information, will help you understanding and identifying those hubs. And then you can go in. And so my way is I start with this big scale, thousands of data network, and I try to identify hubs, and I go into those hubs or pathways. And so the last picture that I showed, where we had those hubs of metabolic pathways that are popping up, there's a subset of 10 to 15 proteins and metabolites that we can now focus on. But we start with a large scale, and we then go down and have something that we also as humans can interrogate. But my appeal to the audience would be really, use the computational tools that are there. When you get tens of thousands of data points, don't try to summarize them in 3 metabolites or proteins that go up and down. Just, it's rough. I'm learning this now, trying to get the past editors and reviewers, but I think this is important, because I think also the audience, we as scientists and readers, we can understand that type of data. Biology cannot be just reduced to 3 or 5 data points. It's more than that. But yeah, that is right now my take. I'm not sure if this is going to stay true for the rest of my life, but this is kind of right now what I'm doing. And there are a bunch of tools available. I'm happy to share the tools that I use to generate those figures, and they're also cited as much as possible in the bioarchive in the paper, and I'm happy to share that with anybody, because they're all open source available. And Leur.
Thank you so much, Jason and Anya. Thank you so much. Yeah. So we're at the end of the seminar, but we have the Metabolism trainees networking session. They like to stay on with Anya, and everybody's welcome to stay who's interested in this session. But otherwise, we'll see you next week.
Thank you so much, everyone. Bye.
Thank you. You can stay behind, everyone.
So I'm going to make, oh, why can't I do that? All right, I'm changing the host to you, Maruka.
Sure.
All right, okay. Well, have fun. Thank you so much, Anya. I know you're moving, and we really appreciate you made time for us.
Thank you. See you in 2 weeks, I guess.
See you. Yes.
Bye.
See you later.
Thank you. That's really, really very great seminar. I enjoyed the science, the techniques, and everything. So I'm going to send you an email, because literally yesterday, just yesterday, I got a lot of mass spec data back, and I don't know how to analyze this. It's just in Excel file, and I'm starting to learn now. So
no, it is scary, but it will get easier. There are more and more tools out there, and I hope I can provide a puzzle piece and help. The community is large. There's so many people out there who are willing to help, and when you approach a mass spectrometrist, just be nice to them.
Yeah, I'm doing that. Thank you so
tell
much.
Them, tell them they like to talk about their data. That's what I can tell you. But yeah, it's challenging. I totally understand, and it's becoming easier. And so my goal also with my lab is to provide more tools like CardioNet. I hope we will get publicly more available. It is available, but as a database that can be interrogated, where you can use it for simulations, also and as a more out-of-the-box solution. But I will need a little bit of time for that.
Yeah, fair enough. I'm gonna ask you one side questions before we move to the career development of anyone. So you mentioned there's a mutation actually in the enzyme, right?
Yeah.
And since you are interested in the cardio oncology, is there's any kind of a mutation in those patients, and the acute leukemic patients, linked to the cardiac risk?
Yeah, so there is data, and actually the dehydrogenase mutation is known. You can find that in the Mendelian database. It's one of the classic inborn errors of metabolism. It's just not a very large patient population, mostly because I think it's also quite lethal. And those patients, they are known to have cardiomyopathies and also neurological defects. And there's independent of our group, there was a paper from Bekelen's group that came out that had they did a global knockout of IDH. And those animals also had both cardiac and neurological phenotypes. And so it's always going hand in hand. And what is quite fascinating that you basically have gliomas that mostly expressing those mutations, and then you have the blood system, or and by proxy, of course, the systemic effect on cardiovascular system. And because cancers in the heart or in muscle are quite rare. Myomas are one of the rarest tumors that you can find, but the metabolic effect is quite fascinating. And as the heart that constantly has to beat and constantly has to maintain a steady ATP provision, this of course has a completely different challenge. And so I find this both scientifically fascinating, but also then again from a philosophical standpoint, but again, different story. Any questions, if somebody has, I'm trying to answer them. Please,
we appreciate your that.
Please be nice to me. The second half of March is going to be a little bit rough on my end, but starting April, I'm back.
Yay.
So, hi, I'm Ada.
Hi, Ada. Nice to meet you.
Nice to put a face to the name, right?
Yeah.
And we actually encourage all the trainees to open their cameras and speak up if they have any questions. But I mean, can you just tell us the story of, how you got your job? And, I'm now in the same boat. Like, I have a K99 and it's been impossible to get a job this year.
Let me get my beer. I'm German, I can say that. Where is the story? Yeah, it's rough. And I think anybody, including myself, who is in that boat, it's not easy. It required a lot of patience, a lot of work, but that's something that when you have a K99, even if you don't have a K99, if you're just in science, this is something that we do on a daily basis. So what did I do? So my story is I started applying in fall 2019, classic cycle, sent out my applications, got like, I think it was 10 interviews or so, something like that. I got a really nice selection of interviews. I was very happy, very pleased.
Was that
I
your
went-
first year of K99?
Yeah.
Mhm.
Yeah. Sorry, time is blurry.
I know.
Yeah, no, that was I was basically 6 months in, less than that. I decided to apply as early as possible because I wanted to. That's my general approach, both with grants or anything. I always want to have a second round. Just I assume that I don't get it the first round, and the first round is for me to learn how the game is played. What do I have to expect? And then I'm going to be so much better the second time. So I always plan in to have multiple revisions or multiple submissions. That's my approach. And so I applied as early as I could, and I spent basically the summer or the months before to tell everybody that I'm on the market. I prepared my package, I had everything lined up, and the response was quite good. And I had all those interviews, and then I was invited to second rounds, and then COVID hit. And I was lucky that was about half of the places I continued to have communications, which occurred during the spring and early summer 2020. And the other half either never responded back or plain told me. And this was a mix of both Ivy League and like mid-tier institutions. It didn't really matter. They all told me like we're canceling the call searches because nothing is going to happen, and the dean is asking us to save money. And so that was it. And then thank you and good luck. And so I was left with a handful of places that would still interview me. And what I then ended up with is that the timelines just dragged on because nobody really knew at that stage what was going on. And are they going to open up? Is COVID just going to do poof and then we go back to normal? And of course we didn't. And I sort of expected that. And mostly because I'm German and pessimist. So I was like, this is not going to go well. And so I prepared for the worst, which is I have to reapply. And I activated my network. And how did I, the short story is, how did I get my job is I activated my network. I talked to everybody that I could. I activated my mentors, which had basically more like life experience type of advice rather than like tangible active advices. But I tried my best to give seminars, write that I'm on the market, keep my name out there with my network that I had, because I was expecting that all the conferences, which also happened, would be canceled in the summer. So there was absolutely no way for me to network. And if they would go into a virtual setting, this would be just a complete mess because people would be overwhelmed already with giving their talks virtually. And so having poster sessions or trainee networking sessions would be almost impossible. And so I basically then through this networking had the chance of giving additional talks and started applying and contacting other places as much as possible during the entire last year, and then reapplied in 2020. But at that stage, as I reapplied in fall, and I had 6 places that I could apply to. The year before that, I could make basically a selection. I was very targeted in where I applied to. I think I applied to more places than the regular people would do, but I'm
How many, if I can ask?
I think it was like something between 20 and 30.
Mhm.
I just, because I wanted to be also open-minded. I'm not particular based on hung up on a geographical location, and I wanted to have a good mix between so-called like high-end IVR1 institution and then anything that is perceived medium, because I truly think that you don't know how a place is until you know, and until you have actually visited them and saw, met people. And there were scientists in some institutions that I cherish, and they are very, very good. They're doing really good science in my particular field. My criteria was always to go for like, where do I set myself up best for my next career step? Where can I realize my work, and not like I'm gonna make it work. I did not want to have that. I did not apply to anything that doesn't have a medical center or medical school, because I want to do translational research. And so even if there may be that great research institute somewhere with a fantastic reputation, I would not apply to that, because I would be just in an island and I would be misplaced, and they probably would look at me and think the same. So it's a waste of time. And that was the same approach I applied this time around. And it was just luckily that all those places that I applied to had essentially also a medical school attached to it. And I heard back from about three or four. No, where's more? I had 8 places that I could apply, and I had 5 interviews. And so I was again really, really lucky. But then Cedars had already interviewed me, and I was sort of ahead with Cedars compared to other places. And they made me an offer in November, December. We negotiated, and I was then at the stage of having the second round of interviews, or third round of interviews with other places. And I just decided to just call it a day. I was happy with Cedars and what they put on the table. I'm happy with the place, and just decided this is going to be my new scientific home. And yeah, the only thing that I noticed, and again, this might change because everything is right now changing and nobody really knows what's going on, is that, in my opinion, like I would say 99.5% of the places that I applied to did not change the way they conduct their academic search. They did the same timeline of like, you do the first interview, then there's a month or 2 in between, then you get the second round, and then maybe a third round, and then you negotiate, and so on and so forth. There has been, they personally also told me that their timeline internally hadn't really changed. They were doing the same thing. The only difference was they were doing it remote, and they were doing it with social distancing. So I had the funny thing where I would travel to a place, and then they put me into a conference room, and I had Zoom calls with people. And I was like, okay, I could have done that from home. But yeah, the advantage is you can see the place, and that is a huge advantage. And so it is difficult on that end. But yeah, that's my story, I would say. Questions?
Yeah, so I'm going to follow up with this question. So just one step backward, I saw that you have 2 projects going on. So did you plan to have those 2 projects with you as a postdoc? And how did you start planning for this? One in muscles, yeah. The
I actually have
cardio-oncology
more than
stuff.
that. So
okay, that's good to know.
Yeah, no, I didn't plan any of that. I came from Germany so I had done Cardionet, and I wanted to do experiments. I wanted to generate my own data and understand. So the project body that I was interested in is how different forms of nutrients are metabolic. Inhibition, inbuilt errors of metabolism can lead to changes in the energy substrate supply in the heart. And that's how I came to Houston, because I wanted to use the working heart perfusion system. I wanted to learn it because for understanding flux changes, this is a perfect experiment to set up. You're neither in vivo nor in vitro, and you can at the same time control everything, which is great. in particular, when you then down the line want to do computation analysis, where you need to have a bunch of parameters and they need to be determined, or they have to be measured in a controlled setting, which you would not always have. And so while I was here, we were approached by Peggy's group, and they had that data. They made this observation, and I was like, I have some ideas. And that's how it started, and that's how we started collaborating. And so one thing led to the other, and I built my projects from the data. And I always say that when I build a project, I follow the data, I follow the numbers. And
sorry, was that your postdoc project, or was that a project you negotiate with your postdoc mentor? So
it wasn't
you
really
see, it's
a
difficult.
negotiation. I
Okay.
told him, like, hey, I want to do this. I think it's great. I think it's a great project. And I guess he is open-minded when it comes to that. He's very, very open-minded. He likes science. And when the data again pushes us in a certain direction, he will follow it. And that's how he is. That's how he thinks. And then I started trying to get funding around this for my project. But it was really like, I was passionate about the topic. I was passionate about the idea, and I wanted to make it work. And since I came with my own money anyway, I was like, yeah, I don't cost you anything. So let's do this together. But this is also not the way he thinks. But what I like about fellowship awards or anything that gives you money is gives you this independence also to say, well, you and I, we actually have a collaboration here. And this is how I went into my postdoc, which is I learned about what a postdoc means in the United States while I was already here. I started doing a postdoc without actually knowing what a postdoc is. It's completely different, and I would never advise anybody to do it that way. I hadn't seen the lab, I had an interview, like I had heard him on the phone, but I've read all his publications. And I said, I want to learn this. This is why I come to you.
I exactly did the same as you. So I came from England for project, not for a postdoc, for project. So
yes.
like,
and I think if you start thinking around projects, then it becomes easier to work together and also negotiate or talk with your mentor about how much time do you want to allocate to this or that. And in general, I think the observation that I have made, if it is promising, if you can show them something that is promising, there are rarely any mentors, any PIs who will not tell you, yeah, follow up on it. But they might put constraints, and then it's your job to be organized about it. And so have your project plan, make sure that you manage your time correctly. And for that, you can get advice from others. But I think that was the part. And then when we saw that this could be a little bit more, and there are more questions that came up that were exciting, and you're either deep down a scientist and you go for it, or you're not. And if you get pushback on that, you're in the wrong place. That would be my advice. And then you should change positions and just but I think in projects. And so I have from that developed the K99, and I wanted to have a mechanism to stay and build my lab because I liked it more. That's why I also didn't continue being a medical doctor, because I was just too excited about the science and I wanted to do more. And I thought, I was just like, if I go back, this is just going to be dormant, because it would take me years to be able to get to that stage again. So I was like, I want to do this now. I'm just too eager. And yeah. And so from there, because the lab did not have this project, and because I developed it, I wrote those grants. It was then also sort of straightforward when it comes to differentiating myself from the rest of the lab. I already came with a unique background. The computational systems biology background did not exist in my lab, and will not exist when I go back somewhere else. And so my PI and Dr. T, we always looked at this as a collaboration. I brought something into the lab that was unique and that would help them. And I got something out of it. And I think this is a perfect level for collaboration. And that's how I also personally look at postdocs myself. And so I have quite high expectations for postdocs because this is how I think about it. I really, you're not a student anymore. You are, if we would go and work for Pfizer, they would put us on a project. And this is similar. We just don't have the structure in academia that would kind of allow us to grow into this. We are not given the project management course or like the software coming with it. But essentially, this is what we're doing. We are project managers. And yeah, I now have the terminology. I did this LinkedIn course during COVID, and now I have all the terminology for it. Highly recommended.
Okay, we will do that then.
No, it's fantastic. It's
really?
great. It's like it's essentially what we are. And
right.
yeah, it's
this is where you keep yourself. It's like marketing strategy, right?
Yeah.
Marketing your science, marketing yourself.
It's what we're doing. And there are some institutions who look at scientists that way. And I mean, what are indirect costs other than we're renting the lab from the institution? That's essentially it. And we're just giving it a different term, but we're still giving them money. I'm learning, and depending on where you are, you have higher rents. And some places it's 33%, and some others is up to 60. So yeah.
It's 70%.
Oh, there you go. Yeah.
It's great. 70% is really fantastic. Really fantastic. So I'm gonna ask you, how do you prepare yourself before you go into job searching? Other than writing the documents, is there any course that, or is there how you prepared yourself? You know, it's like you're going into the war. You wanna get all the instruments. So how did you approach that?
So if your institution has a career development office, talk to them.
We don't.
Yeah, we don't either. I didn't have it. I just looked on the websites and I thought it would be nice to have that. There are some institutions, UCSF is one that has some material online. So what I did is I bought literally the book The Professor's In from Karen Kelsey. I think is her name. And I watched also some of her podcasts. I think it's not anything that works for you. What worked for me, again, is a variety of things. I talked to my mentors. Half of them told me they can't help me because they haven't interviewed people in a while or had been on the job market in a while, so they have no idea how it goes. The other half had postdocs on the market and they could give me some advice, but what you need to understand is each mentor has a limit. They come from a different background. Are they male? Are they female? What age range they are? Are they person of color or not? All those things. And that drives also the validity, or that defines how valid is their comment and their advice and their life experience. And so you need to kind of listen to it, but don't take it with as a dogma or so. Like, weigh it, and they can be wrong, and some people were wrong, and some, when it comes to COVID in particular right now, everybody will be wrong. Every experience that you will have will be unique because your committee, the search committee that and your mentoring committee, they're all going through this for the first time. And what I noticed when doing short talks via Zoom or interviewing via Zoom, committees, at least last year during that cycle, and probably for another year or two, will not know what they actually want, but they know they don't want this or they don't like that, but they're not going to be able to voice it before they actually have seen it and done it and gained the experience themselves. So anybody who comes and tells you like, don't do this, you need to do that, take it and put in your own judgment. And then if you have an interview, ask the chair of the committee for specific question, like criteria. What are they looking for? What do you want me to do? They sometimes give it to you, sometimes they don't. And if they don't give it to you, it's not because they're mean or they don't like you. It's because they haven't actually really thought about it and they don't have defined those things. This is what I noticed. And if you have the chance, visit the place and do it in person. It's going to be so much better. Every place where I had the chance to visit in person, I did it in person because it's so different, in particular for a shock talk. Giving a seminar via Zoom, yeah, fine, it's okay. But a shock talk is different. And there are people who are really good with shock talks using presentations, and some people are not, and they prefer doing it on a chalkboard. And I'm one of those people. I'm really good on a chalkboard. I'm not good in doing a presentation in a shock talk. It's just not the way how I interact with my work. I'm really dynamic. I walk around, I draw doodles, I have my schematics, and you can't do it effectively with a screen. The main reason for that is you are just talking into the void. And what most of the committees don't fully understand, because they have never done it, it's incredibly difficult to describe your science and future projects if you have zero response and you're just having this computer screen. And maybe your cat walks by or your dog, and that's about it. And so if you can have, and I haven't tried it beyond one talk, but if you can have somebody in the room, or you can have sort of a fake audience, it helps so much. I had to do a shock talk in person at a place, but you basically, again, they put me into a conference room and then everybody was on Zoom. So I had basically a panel with like names, everything black, and then they were all muted. I saw like 40 of those on
No camera.
No camera on from anybody. And then I was there and I was supposed to do my project. And then they had like one of those fancy cameras. I was following you around and it was great. They had done a huge effort, but I was just in an empty room. I had no audience. It's like the musicians. Everything that you hear from musicians right now about the difficulty of performing during COVID applies to us right now in interviews and also in scientific setting. And whatever they came up with, my cello teacher and I, we talked about some of those things. And so you need to either be really, really mentally strong and have this natural ability of like imagining an audience, or you put an audience into the room. And what the place did, which I thought was really, really great, they had two people in the room. And one was the organizer and then another faculty member, and they were socially distant. And that made a world of a difference because you got sort of a facial expression and there was a communication that is happening. And that was not just in my head.
You can read the faces if you can have that in person. Yeah, whether they're like interested. Yeah.
Yeah, because unless you are used to it, and I'm not that type of person, I do not blab all by myself about my projects without anybody being in the room. Some people do that. I generally do not. I don't use my voice when I think about my projects. I don't talk to imaginary friends. And so it was very rough for me. And so what I started then doing is booking a conference room, even though nobody was there, and then maybe putting one or two friends in there, and they give you facial expressions, and they can tell you when you're offline or something, and that's a teeny trick. And I would use that. I would highly recommend doing that. If you are forced to do a short talk via Zoom, and if you can use a whiteboard, try to find a technical solution. If you cannot, try at least to still have a person in the room that listens to you and gives you that expression, because it makes you more confident. It takes a lot of energy talking for an hour and being confident and excited about your projects. And that is actually what will be judged also. They have already judged you on, do you fit? the interview is there to understand for you also, do you want to be in that place or not? And that's very important to realize, even if it's COVID. And so what was, again, the scary thing for me, of course, was that I took a job offer and I didn't had done all the other interviews, and I didn't had given those other, like the second round interviews, and I didn't give the other places a chance to give me an offer, which they would have done. But I made my decision beforehand because I could see the timeline, and I had done for myself the decision that I do not want to wait another 4 months. And this is what would have happened. And this is the other thing to keep in mind, is that a lot of the academic searches are still being conducted as if we would not be in COVID. And this is the sad part. And I don't really understand it. I hope if I ever going to be in a position of power to design those things, that I'm not going to do that to anybody, because it's one of the worst things. And I was just lucky in the sense that I found the place that I liked. And I made, again, I'm also the type of person that I ask myself, what do I want? I had that defined. And then I just objectively tick the boxes, like, are those things fulfilled? Yes or no? And then it's like with apartment searches, you can go on and on forever to find your dream apartment that's the best in the world, and everybody is going to envy you for that, or you settle maybe for 95% of what is on your list. It's still going to be 95% of that is great, and maybe the 5% that are not great or that are not on your list or not that fantastic are actually not that much of a deal breaker. And so I define what is a deal breaker for me, what is really a red flag, and the must have, and anything that is in the middle. And so I had those categories, and I knew exactly what I wanted and what I didn't want. And when I found that place, I said yes. And I talked also with all my mentors about it. I talked to a bunch of people until I couldn't talk anymore and I couldn't discuss this anymore. But I got all their input. And then in the end, it was a gut decision, and time will tell. But the thing is also important to know that no matter if it's a pandemic or not, you can always change those decisions. Nothing is final. We can change, we can get another job, we can leave academia. It's going to be okay. You're still going to be a scientist. You're still going to be...
You're still going to be you, right?
Yes, exactly. There's so many, again, I like this analogy when it comes to musicians. There are so many different types of musicians out there. You have soloists, you have orchestra musicians, you have people that only do chamber music, some others just play soundtracks, and they're all musicians in the end of the day. And you need all of them. You need the cellist, the second, the third, and the first cellist. And then you need the soloist. Otherwise, you're not going to have a nice concerto. And all of them are great. And so it's just, what do you like and what do you want and where do you have the opportunity and what makes you happy? And that's how I look at this. And so I can do what I want to do. And after that, after 3-5 years, I can revisit that, and Cedars or my new place can also revisit that. Again, it's a collaboration.
It's two-way relationship.
Yes, you're in charge, and it's not like the doomsday scenario. But what is difficult, and this is where it's really, really unique, is that you need to have this type of motivation. And what I found very, very challenging sometimes is to motivate yourself. In particular for all the Zoom calls. And what was very, very challenging is that despite the fact that it's supposed to be easier, the Zoom interviews led to me having basically visits at places over the course of 2-3 weeks. And that was very challenging. That was actually the most challenging part. And
so you can keep yourself focused on one thing.
Yeah,
right?
and still do your work, because you don't want to be like working nonstop on interviewing, but at the same time you want to, because you really want to have a job. And so I think what is also what was empowering for me to notice is that you can take charge of your calendar. define what experiments or would work outside of you trying to find a job you need to do. I started scheduling everything down to my lunch break. And I put that into a very detailed calendar so that I could also tell the institutions, I have a meeting, there's a conflict. You don't need to go into a specific, you don't have to make it work for everybody. You have that right. And if they cannot make time for you, you really need to ask yourself, do they actually want me? It's also a test. And because then you can save that time. I mean, we have so much stress as postdocs during COVID. You don't want to stress about your time and on top. And so I didn't do it in the very beginning, but then after I took also a LinkedIn course, I found out that you can do this and it works actually really, really nicely. And it empowers you and it gives you a little bit more control and less stress about like, oh my God, I need to meet that other deadline. I need to write this review. I need to write this rebuttal for that manuscript, or I need to get this like other grant out or cost extension for my grant that exists. And then you are also more focused on your actual interviews, because people still gonna judge you after this same criteria as pre-COVID. They're not gonna give you a slacks on that. And the lived experience of postdocs versus faculties was right there. Like people looked at me sometimes and were not understanding at all. I mean, I'm in Texas. We are basically open since April last year. So there was a month that I was not in the lab. After that, I was on the full, like back in the lab. Yeah. And we don't have screening stations in front of the building. Nobody screens anybody here. So I'm like, okay, great, fantastic. That's not stressful at all. So
I kind of the same situation. So I'm Shreveport, so it's not that far away from Texas, Louisiana. So
yeah,
we didn't stay in home as other people. We worked full force, and
it's great. It's great. You can still get things done, but also the labs are closed, so you're still working alone. You're having social distancing protocols. So I was alone the whole day in my room, and yeah, it's okay for a month or two. Now we are at a year, and I don't. I think it's important to understand that and also vocalize this and remind people. And you can do that and understand that your experience has been different from the faculties. And also understand, and I think this is realistic, if I would go on the market now, and if I would have a trainee, I would recommend to them also to either seek advice from career counseling that is available at the university, or seek advice from professional counselors. Because you need to have somebody else than your friends and your mentors to look at you and give you an objective opinion about your package, about you, about how you present yourself, and schedule those things also via Zoom. And I think that can help tremendously. Yeah.
Great advice.
Yeah.
Thank you so much.
I wish I would know that before.
Yeah, I know, right? We're lucky to have you. We are so much lucky to have you. So
yeah, use that. Absolutely use it.
I got all the notes, so we're gonna follow that.
Yes, and if you have to pay a little bit of money, I think it's well invested. A friend of mine did that, and it worked out quite well. And because with that money, you're buying yourself not only material and advice, but you're also buying yourself time, because you do not have to do it yourself. You're going through this for the first time, but everybody has been sort of going through this for the first time. So there's knowledge out there. It's just that we cannot always harness it. And one way is career development offices, and another way are those consulting companies. And I would, there are some that specialize on academic searches. And with the market being so fierce, you have to use it. I applied to positions where they had literally over 400, 500 applicants.
True.
You cannot possibly, like, you're being selected based on very, very, very stringent criteria, and one of them is being, have you published? Do you have a grant? Period.
100 publications.
Yeah.
Yeah, number of publications, always an issue.
Yeah.
Something you cannot control.
No,
True.
and I would also, even if it's not very popular, I would definitely advise you also to look for other fields. I did look into industry, and I would have applied for it. It just worked out for me. But a friend of mine was applying for it. It worked out for her perfectly. She's very happy in that position, and there are positions out there, in particular when you are now with a different administration and the way they're looking at H-1B visa, but in particular when you're an American citizen, it's an option to look into. It's there's great science that is being done in industry, and I would not use this as something that is negative. And so, do you want to be a professor with like I will rest somewhere in with like pine trees and all those things and nice garden, or do you want to be a scientist? And you can be a scientist in various different positions and various different institutions. And so I explored that as well. I also considered going back into medicine. I had that as an option. So I had plan A, B, C, and D. And yeah, it was there. And I just, my plan A worked out. And or actually it's plan B. Plan A was I go back to medicine.
Medicine.
Yes. And so so I got lucky on that end. But yeah, no, right now, I mean, it's numerically not possible when you look at the data and compare it to 2008. How long was the academic surge disruptive after the crash at Wall Street in 2008? Now we have an even worse economic situation. There is no way that this is going to recover faster. So at a minimum, it's going to be the same as post 2008. And it took 3 years. And yeah, 3 years. And then people slowly got back to the baseline before. And so now I don't even have an idea what timeline we're looking at. It might not go back at all. And when you look at very small institutions and the junk positions, I think that market completely evaporated. And so there are studies out there and numbers. And again, I think the team from the professors in there kind of following it. I'm not sure if there's another source, but they kind of present that, and they're quite good in gathering all this information, at least from the humanities. And I think it's not that much different from the science.
You know what? I wish we can meet in person. Seriously, we can have a lot to talk about.
Yeah. No,
so it's a shame, though. It's a shame we're doing that through Zoom. So we are so much grateful for you and your time, the advice, everything that you give us. So thank you so much. And we will follow up absolutely by email. Absolutely, I'm gonna follow up with you. A lot to ask. So
yeah, it's the one thing that I really would like to convey to everyone, it's not you, it's I put, like, there was one search where I put so much energy into it, really did my best, and they still didn't go for me, and they criticized the way I was presenting. I was like, whatever. I'm doing this via Zoom during a pandemic, and I'm working full time in the lab, so if you don't want this, it's fine.
Very true.
Yeah. So have different options lined up, and be aware it has nothing to do with the quality of your work, you as a person, and what you transpire also in those meetings. Not your failure. I think the system right now is failing us. It's the other way around. Yeah.
Very true. So
that helpful
thank you
note.
so much. Yeah, very helpful.
Yes, lots of energy.
Cannot say anything. Yeah. It's the
Thank
system
you so
is
much
not
for
you.
your time.
No,
Thank you so much.
You're very
Thank
welcome.
you.
Yeah,
And
please
we will.
Yeah, and congratulations.
Absolutely.
Thank you so much. And yeah, please reach out. You find me on Twitter. Just connect with me. And I'm gonna still have access to my UT email. Otherwise, it's my very long name at cshs.org. So I'm gonna be at Cedars, but there hopefully going to be a website with my name on it at some point too. So you can find me there and just reach out. I'm happy to help and tell you what I felt like doing this last year and a half, and yeah, help you as much as I can. But yeah.
Thank you. Thank
Good
you so
luck.
much.
Good luck,
Thanks.
everybody.
Have a nice
Thank
weekend.
you. Have a nice weekend. Thank you for
You
your
too.
time.
Bye.
Bye.